morning, children. Now we are dealing with the lesson, the story of microorganisms. So in your class, that this textbook contains two parts now. That is the story of microorganism part 1 and the story of microorganisms part 2. The story of microorganisms part 1 consists of the different what are microorganisms, what are the characteristic features of the microorganisms, and different microorganisms, how they cause different diseases. So these are the content that is given in this part 1 chapter, and the next chapter it deals with the scientist who has developed this about the microorganisms, the discoveries, inventions of different vaccines to these diseases that are caused to their microorganisms and so on. So we'll learn detailedly one after the other. Now we'll move to the story of microorganisms part 1. So what are microorganisms? The name itself, you first should know what are microorganisms. Already some of you are aware of this word, isn't it? So the lesson is about the story of microorganisms. Microorganisms, the living world, the earth consists of so many organisms. In fact, these organisms are very minute to so many larger organisms are present on this earth. So the microorganisms are the minute, microscopic and invisible to the naked eye. So the organisms which are invisible, not visible to the naked eye, directly we cannot see them. So the organisms are the organisms which are invisible to the naked eyes are called as microorganisms. If you want to know about the microorganisms, if they are invisible, how can we see them? That is the question arises. So these organisms were first observed by the Antony van Leeuwenhoek. Before him, we were used to know only about the living world, but that visible world only can be observed. The living world, which is visible only, is known to us. Before his experiments. After this, that's by Antony van Leeuwenhoek, who was the founder of microbiology or the father of microbiology, van Leeuwenhoek. His period was between 1632 to 1723. So it showcases that Antony van Leeuwenhoek worked on the different microorganisms. How did he do so? He has invented the microscope, that is the single lens powerful microscope. He worked with his single lens powerful microscope, which magnifies the objects 300 times more than the object is. Is it clear, children? So, Antony van Leeuwenhoek is a scientist. At first, he was a fabric merchant. He was working out with these various glass lenses. While doing so, his curiosity and interest to work about the lens made the advent of the several powerful microscopes. So, the creation or the discovery of the different microscope and their inventions made possible through his curiosity about the various lenses, observation of various organisms through his lenses, which he used. The microscope has a capacity to magnify the object that could be seen out of 300 times more than the object is. Is it clear, children? So he has paved the way for the observation of different organisms, minute microscopic organisms, that which we cannot see with the eyes directly, that is, that which we cannot see with the naked eyes. They can be observed through the help of the microscope. He, in the year 64, I think so, it's, 1674, it's 1674, he was called as the father of microbiology, since he had worked about the microorganisms. So he was working about the microorganisms. The study of microorganisms is called as microbiology. Biology, you know, the study of life. Microbiology is the study of microorganisms. Study of microorganisms. It's called as microbiology. So, Antony van Leeuwenhoek was called as the founder or the father of microbiology. Microbiology. His works about the microorganisms created or paved the way for the knowing or the knowledge of various different microorganisms that we know today. Okay, children, so what are the different microorganisms that he observed and the observations of the other scientists of different microorganisms we'll see now. So he has observed the microorganisms at first as bacteria. Bacteria was observed by him in the year 1678. He named it as bacteria. Before that, he gave the name as animalcules during his observation through his single lens powerful microscope. Okay, children, so before that he named it as animalcules and later it was named as bacteria in the year 1678. Antony van Leeuwenhoek. So the different microorganisms that he studied are bacteria, as you know, as usual. Next, coming to the protozoans, fungi or yeast, protozoans, amoeba and all, they have studied about it. Protozoans, red blood cells in human beings. So various microorganisms and also the different cells he had studied, Antony van Leeuwenhoek, and give the information about them which are minute and microscopic and cannot be seen with the naked eye. So we'll see about the microorganisms that are being studied by different scientists and what are they and how are they absorbed. Microorganisms. First one is bacteria. The different forms of bacteria as such are coccibacteria. Write these names and note it down, ma. Coccibacteria, bacillus bacteria, lactic acid bacteria, that is called as lactobacillus. Lactobacillus. So coccibacteria, which are spherical in shape, bacillus bacteria rod shaped or they are in rod shaped, and lactobacillus are the bacteria present in the milk which helps to convert the milk into curd. So these are the bacteria that is that's helpful and useful to us in different ways. Some bacteria are useful and some are harmful. About their useful bacteria and harmful bacteria, I will tell you in the further classes. Next, see here now, we will see about the organisms, microorganisms now. And note these words are very new for you, and the terms also very new. You have to know how to pronounce them and also rewrite them in your notebook so that you can easily pronounce and write well. So the next one is about the algae. These are normally found in the oceans, seas, ponds, rivers, and also in our tanks also. Obviously, when the water is stagnant for a few days, you can see this formation of the algae there. So the diatoms, etc. are found over there. Chlamydomonas, Spirogyra, diatoms, spirulina. See here, children, Idagonium, Serratia. So these are the different algae varieties that are microscopic and found under the microscope itself. They can be found microorganisms that can be observed only under the microscope. Next, this is an algae, isn't it? Note it down. And the third variety, I wanted to tell you, before that, you write down. Next is fungi. Aspergillus, Penicillium, bread mold. So these are the fungal varieties. Hope you have understood and know how to pronounce each word, and you also know about the bread mold, the mold that is developed over the bread on the moist bread when kept for a longer period. Fungus formation takes place that you are aware with, isn't it, children? Next, protozoans. I told you in the previous class about the amoeba, isn't it? So they are unicellular organisms. So minute and microscopic and can be observed only under the microscope. Slipper animal cure, that is about the paramecium. So these are the different protozoans. Verticella, bell-shaped it is. You have to identify the pictures of each. So they can be asked in your examination purpose. So you have identification of the picture is very much essential for these microorganisms. Next, coming to the microarthropods. Arthropods. Arthropods, the name itself tells that they are insects. Microarthropods, micro, very, very smallest one which cannot be seen with the naked eyes directly. So these are the insects. They are very, very smaller insects. They are cyclops, already called as cyclops, Daphnia, cyclops, Daphnia, scabish mite, and eyelash mite. So these are already given for you in your textbooks, and you can observe them, and you can verify each picture and know and identify them easily. So take your textbook page number 29, children, and observe each bacteria, fungi, algae, protozoans and microarthropods. So these are the different pictures of the bacteria. Bacteria, Bacillus bacteria, Lactobacillus, which is present in the milk. This is the algal varieties of Chlamydomonas, Spirogyra, Diatoms, present in the seas and oceans, Spirulina, Erigonium, Serraceum. And the next is about the fungus, Aspergillus, Penicillium, bread mold. See here, Penicillium, bread mold, Rhizopus, Aspergillus. Next, coming to the protozoans, amoeba, paramecium, verticilla, microarthropods like cyclops, Daphnia, scabies mite and eyelash mite. So these are the different insect groups which are very minute and microscopic, and how they can be observed through the experimental model we'll be seeing in the next class. Hope you have understood the lesson completely up to here about the introduction of microorganisms, who invented them, who discovered the microorganisms, through which instrument they discovered this microorganisms. Everything you have noted down. I think hope you have understood well, and in the next class we'll proceed with the microorganisms, how to do the experimentation over these microorganisms. Okay, ma? Have a good day. Thank you.